we have that is related to gene sequencing. We can have again varieties in this. It can be whole genome sequencing, that whatsoever genetic material we are it is present in the cell, that is whole genome sequencing, and in that everything whatsoever genetic material is there. We have seen that some of the genetic material is present in the mitochondria also, and some of the genetic materials are present elsewhere, ribosomal, and next is most of the genetic material is present in the nucleus. So at various places whatsoever is there, all type of DNAs this are there, and if we want to have another only specifically DNA sequencing related to nuclear genetic DNA, then we'll have another way. But then why it is there? So here it process of determining the complete DNA sequence of given organism, whether it is a plant, whether it is an animal, whether it is a human being. For that whatsoever kind of genome is there at given time, at single time, whole sequence will be there. So all kind of chromosomal DNAs as well as all the DNAs which are contained in mitochondria or at any place, other place, as I told you, example in plant it is in chloroplast. So all these DNAs they are checked, and this sequence is verified. This is called as a whole genome sequence. Now it was basically thought as a research tool earlier, but nowadays after this recent in 2014, people are trying to relate it with personalized medicine. That is because we have seen that there are many diseases, they are because of the genetic problems, and this genetic problem if we can be determining, then we can understand what kind of problem the people are having and what is the basis of this problem. And with this if we can try to give the either personalized medicine, or we can try to correct the genomic sequence, it can be helpful and it will be given a better one. So whole genome sequence, it can be important tool in the guiding the therapeutic intervention, and therefore these all details you are studying. The tool for gene sequencing is at SNP level, that is single nucleotide peptide. We have seen that these are various SNPs are there, where there is a change in the single nucleotide peptide is there. Because of change in the single nucleotide, one nucleotide here or there, that is going to cause change in the protein, and this is causing change. We call it as a single nucleotide protein. That is also used to check or pinpoint the functional variants which are responsible for that kind of phenotypic problems. We have seen any type in any type of change in the genotype or genetic change is reflecting as a phenotype change. So we are going to check what are the genetic changes reflecting into various phenotypic changes. And now this, it can be diseases, it can be disease susceptibility, or it can be drug response. If you remember, I had told you many times that in 5th year you have a syllabus in clinical pharmacokinetics, where you will be able to pay attention on the topic that is pharmacogenetics. You might have studied it earlier in pharmacology or even in biopharmaceutics, but here it will be given more details that how variation in pharmacogenetic features will be responsible in the changes in the various drug response. This will be pharmacokinetic or pharmacodynamic. So this is why you are studying this. This is why you need to study this all details. So this genome sequencing should not be confused with DNA profiling, and the only determines the likelihood of the genetic material into it. So we are having whole type of genomic sequences that will helping us to understand. And as of 2017, there were no complete genomes for any mammal, although human genome project was conducted long back, but still complete genomes were not available, including humans. 4 to 9% of the human genome, mostly satellite DNA, has not been yet sequenced. Although majority of DNA is known, sequences are not, but 100% is not known. Let us see various things. I will not go for history about it. Actually, all scientists are trying to understand what kind of genetic material are present in various bacteria, plants, since many days, then various animals, particularly laboratory animals, what kind of genetic situations are there, and Hello. Is it Sahil is not able to hear, but whether it is there with everyone? Others are able to hear?